Hello and happy Friday. Let's go ahead and start off by taking out your homework from last night. So that would be the transcription section of the Breaking the Code Ditto. Starting off with your first DNA molecule, building the complementary RNA molecule, you should have gotten AUG, GCC, UAC, GGU, CUA, GUU, and UAG. Breaking each into codons, as you can see demonstrated on the screen. Moving on to molecule number 2, the complementary RNA strand you should have gotten were AUG, CCC, CCG, CAU, UGG, UGU, UGA. Moving on to strand number 3, it is AUG, GAC, AAU, UCG, AUG, UUU, and UAA. As a little side step, right, really we need to evaluate where this process is happening and what's happening. So let's just kind of talk about this for a sec. I'm asking you, if I'm going from DNA to RNA, what exactly is this step called? Take a moment here. You're right. It's transcription. Since the transcript or the mRNA is actually being created, this step is called transcription. And where does it occur? You're right, the nucleus. It has to, because the DNA is the ultimate guide for this process. Excellent. So I'm going to proceed on in your notes, and at this point you can open up your note packet to page 10, titled Post-Transcriptional Modifications. So like I've already mentioned, the process of post-transcriptional modifications, so this is something that happens after, that's what post means, transcription, the process where we make mRNA, and it's a preparation step. The cell environment is quite harsh, and that mRNA needs to be protected. If it's actually not protected, it can be destroyed, and then, unfortunately, the protein produced from it would be mutated or altered. So a few things happen in this process. The mRNA molecule will receive a methylguanine cap, protecting the front part of that RNA molecule. Step number 2 is the other end, or the tail portion of the mRNA will receive a poly-A tail. Now, poly means many, and A actually stands for adenine, so we'll see in a video that it's actually many adenine molecules added to the end. The third step in the process of post-transcriptional modification is the portion of the mRNA that is not needed or doesn't get coded for is actually removed and sliced out. And so there's portions of our mRNA molecule that are DNA that is not needed. They're referred to as introns. Now, I really hate to call it junk DNA, but your textbook actually refers to it as junk DNA. In reality, it's not junk. It's DNA that is there to protect from mutations. There's a functional purpose why there are additional nucleotides added to the DNA. And that's so that if there is an error in the process of replication or in transcription on these portions, the protein would not be affected by it. It's a really great evolutionary protection against mutations. The portions of the gene that will actually code for the protein are referred to as exons. So these are the expressible sections of your DNA. So in this very last step, the introns will be removed, and of course we use enzymes to do that because they speed things up. And the introns will be removed and the exons will be glued together. Now, of course, this happens in the nucleus because this is directly after transcription, but before the mRNA can leave. I'd like for you to go ahead and pause the video here, and please watch the video. This post-transcriptional modification video, processing of gene information, prokaryotic versus eukaryotic. Pause the video here and watch the video. Welcome back. So that video is a great depiction of the 3 steps of post-transcriptional modification. You saw the methylguanine cap being added to one end of the mRNA, the poly A tail to the other, and the removal of the introns and the unionization of the exons. After this step, the mRNA is prepared to move on out of the nucleus, and it's going to have the introns nicely glued together. We can see the methylguanine cap on this end, the codable region of the DNA, and the poly A tail on the other end. And now this mRNA, following post-transcriptional modifications, is prepared to leave the nucleus. It can now move into that harsh environment and move into the step of translation. So this is the step where we're going to see, the molecule of mRNA being read by tRNA and translated. So when this actually happens is when the protein that this gene codes for is needed, and it needs to be made. And what's actually going to happen is the mRNA code, which of course originated from DNA, is going to be converted into the language of proteins. And we know that the monomers are amino acids. So the tRNA is the molecule that is able to read the language of mRNA and convert it into amino acid language. So the tRNA will carry the anticodon, and the anticodon is the opposite or complementary to the mRNA codon. And where this is going to have to happen is within the ribosomes. Now remember, those ribosomes can be free-floating in the cytoplasm or embedded in the rough endoplasmic reticulum. And this is happening so that the protein that's necessary to be produced for the cell to properly fulfill its function needs to be created. This is a great image of tRNA, which we've seen once before. Here we see the anticodon on the bottom. We see the very stereotypical characteristic of the folded upon itself with the amino acid on the top. I want you to pause the video, please, and we're going to move on to this awesome translation video. It's this protein synthesis second link right here. Take a moment. You might want to watch it twice in order for you to get a good grounding on the process of protein synthesis. So please pause the video and watch this second video. Welcome back. I hope that video was a great outline of the process of protein synthesis. So we're going to go through the steps of protein synthesis and translation. The very first portion of this process is called initiation. This is where 2 codons of the mRNA sit on the ribosome, and the first codon will always be AUG. In fact, AUG is the start codon and the complementary anticodon that tRNA binds and brings over the amino acid. The amino acid will always be methionine. So AUG is the start codon, methionine is the appropriate amino acid that goes along with that. And this is how the process of translation begins. Following initiation is elongation. This is the step where codons are read along the mRNA strand, and the anticodons of the tRNA are complementary to those codons. Those complementary amino acids that the tRNA brings along on top of it is actually going to form a peptide bond, linking together those amino acids, slowly forming the polypeptide. Now, 2 codons can sit on the ribosome at once, no more, and it will allow for those 2 codons to bring along 2 amino acids, and they'll share that peptide bond in between. All of the mRNA codons are read in the same way until they reach the stop signal. When the stop signal is reached, the termination portion of translation has begun. And in this particular step, one of the 3 stop codons of the mRNA sits on the ribosome. No tRNA can bind to the ribosome at that location, and the translation process stops and is terminated. mRNA will fall off of the ribosome and will be broken down into its smaller parts, or nucleotides. The tRNAs and the ribosome pieces are actually recycled. Those tRNA molecules are in the cytoplasm and will get amino acids linked to the top, and the ribosomal unit will go on to another strand of mRNA to be recycled and go through the process again. Now, the protein, which has been the whole point of this process, is really important and is actually going to break free from the ribosome. And it's going to be transported to wherever it needs to go. If this is a secretory protein, it will then be secreted out of the cell, of course, carried by a vesicle, and then fulfill its function. If it's a protein that's necessary for that cell, then it'll go to the appropriate location where it is needed. I want you to go ahead. You're going to pause the video again. Here's a great video focused on protein synthesis. I know that there's a lot going on in this particular process, so take a keen focus to detail of all of the important enzymes involved. Please pause the video. I hope you found that video helpful. I know that one's a little bit more challenging than the previous, so if you rely on video number 2, really to outline the process, that's a very good resource for you. So as we look at just a visual representation of this process, showing us that we're in the ribosomal unit, here's my mRNA molecule, my complementary tRNA. We can see the methionine coming along. And this ribosomal unit will hug onto that mRNA molecule. We can see that the tRNAs are coming in 2 at a time on that ribosome and linking together a polypeptide. Remember, a peptide bond is shared between these amino acids. And this elongation process will just elongate the polypeptide strand until we get to the termination signal. These parts are recycled, and then your protein is used wherever it is needed. So there's one key tool when working with protein synthesis that is very important, and it is this amino acid chart. The way that this chart is read is these are codons on the chart, and the first letter of the codon will be found on the left-hand side. So if the codon is AUG, then A is the first letter, and the only row you may look into is this row right here highlighted as A. Saying the second letter is U, well, then you would find where this row and the U second letter row overlap. Now we've eliminated all the options except for what's inside this box. And then the last letter would be G. So I find the third letter on the side over here and I draw it on over. I see that the amino acid that comes along with AUG codon is methionine, and it initiates the process. So let's practice a few here. If our AUG, we know that the amino acid would then be methionine, and the anticodon that would pair with that would actually be UAC. So let's do this next one, CCG. So you've got C in this row, G, now we've got an overlap in this box, and that was C in this row, C in this row, and now we're in this box. And G is the third letter, which leaves us with proline being the amino acid. And the anticodon, that would be what would be found on tRNA, would be GGC. Moving on to AAU, so A would be the first letter, that would be this row. A would be the second letter, we'd be working in this box. And U would be the third letter. We're working with asparagine is the amino acid, and UUA would be the anticodon. Moving on to the next one, UAG, so U is this row. A would be this box. And UAG, I can see right here, is stop codon, which would terminate the process. And the anticodon to that would be AUC. Finally, a little practice, we see GAA. So G would be this row, A would be this letter. Now I can look into this box, and A is the third letter. That's glutamic acid is the amino acid. The complementary anticodon to that would be CUU. You can pause the video here and practice with a few other examples just so you get acclimated to this chart. What I'd like for you to do now is take out your Breaking the Code worksheet from homework and turn to the region that highlights translation steps. So this would be the process by which we would create the anticodons. And so if we are always basing this off of the DNA molecule, then the anticodons would be, of course, complementary to the mRNA molecule. And so I want you to take a moment right now, since you've completed the transcription portion of this, and build the anticodons that would be complementary to the complementary RNA strand number 1. Remember, in RNA, it's A to U and G to C. Pause the video here and complete the translation step.